I'd like to start by thanking Charles and Ray for the invitation here. It's my first Hep DART meeting to come and talk about the immune response in hepatitis B. And I added the qualifier on here, in patients, because all the data I'm going to show is really data that we've now understood in the field coming from chronically infected patients. And I also say that I'm going to, because it is a broad topic, I'm going to cover a lot of different data throughout the talk. So it's going to be somewhat top level, but all the references are there. And all the slides will be available in virology education after the meeting if anybody wants to go back and have a look. So with that, I will get started. And for years, I would say, if you started reading an immunology paper in hepatitis B, this is how the first paragraph would always start. Acute or resolved hepatitis B patients had a robust, coordinated immune response that was comprised of CD8 T cells that recognized infected hepatocytes, B cells that made anti-S antibodies. It was all coordinated by the helper CD4 T cell response. And the chronic infected patients had a weak T cell response that was a low T cell frequency and a lack of effective antibodies. So these kind of very general and vague comparisons between acute and chronic. But fortunately, over the last 10 years, we've made really impressive advances in our understanding of the immune response, and especially the T cell response in hepatitis B patients. And I want to start out by just looking really at the profile of the T cell response in chronic HBV. And Analog made reference to this yesterday, but the clinical definitions of hepatitis B being immune tolerant, immune active, and inactive carriers being a clinical convenience without the immunology data to really support it. And so there was a nice study that was done by the Hepatitis B Research Network and published just a couple of years ago looking at T cell responses across the different stages of hepatitis B. And so basically, they looked at T cells to the different antigens of hepatitis B and also in aggregate. And ultimately, what they found was that there was no significant difference in the level of T cell frequency across the different stages of chronic HBV infection. And in fact, a better correlation of T cell functionality may actually be the ages of the patient. And so there was another study done that looked at immune active patients that were either younger patients that were younger patients that were either under the age of 30 with active disease or in the black bars, or over the age of 30 in the white bars. And what they found was that the patients that were younger had more HBV-specific T cells in their blood. In addition, they also had lower PD-1, suggesting that these younger patients had higher functionality of the T cells that were present. So in addition to what we now know about the frequency of T cells across HBV, we also have a better understanding about the variation in the T cell response in chronic HBV patients. And as I was just asking, related to the age of the mice, so there was some nice data presented at the HBV meetings. Unfortunately, I don't have that data to present, but there may be an evolution of the T cell repertoire within patients as patients age, changing specificities based on the proteins of hepatitis B. And there was also a study that we published almost 10 years ago now looking at how the HBV genotype and the HLA profiles of the population really impact the HBV-specific T cell repertoire. And it might be a bit difficult to read up here on the screen, but in the black bars are Caucasian patients, and in the white bars are the Chinese patients. And what we did was take two classic epitopes that are used to detect HBV-specific CD8 T cells and tested them in these two populations with their corresponding genotypes. And you can see in the Caucasian patients tested with the genotype D peptides, most of these patients elicited a T cell response. When we use the corresponding epitopes in the Chinese population, this T cell response was almost undetectable. So this is due to the genotype, but also the fact that HLA-A2 in the Chinese population has a lot more variability versus the greater than 95% HLA-A201 in Caucasian patients. So what this means is this is really important when we're thinking about measuring antiviral T cell immunity either for research or for monitoring in clinical trials. It also going to have major implications when we think about designing therapeutic vaccines for specific populations. And it's also important to point out that all T cells are not equal. In particular, T cells in the blood are not equal to T cells in the liver. And so this was a nice paper published this year from Malamaini's group looking at the phenotype and function of, comparing the phenotype of function of T cells from the blood compared to the liver. And if you look here on the right, HBV-specific T cells in the blood have a very different phenotypic profile than HBV-specific T cells found in the liver. And this also corresponded with a different functional profile and a different transcription factor profile. So essentially, these T cells don't recirculate in the blood, so what we're looking at in the blood doesn't necessarily correlate with what we see in the liver. And then also, more information on the T cell responses. We know that patients now that lose HBs antigen have a higher T cell frequency. And in particular, the HBs antigen loss in nucleoside analog treated patients, so a very significant recovery in T cell frequency to the level almost found in what's observed in acute and resolved hepatitis B patients. And then there's also, it's very difficult to read, but in spontaneous HBs antigen clearance, there's also higher T cell responses, although not to the level we've seen with nucleoside analog clearance. But the issue here with this study is it's cross-sectional, so it's kind of a chicken and egg. We don't know if those patients cleared S antigen because they had high T cells, or if those patients have high T cells because S antigen is gone. But nonetheless, there is the correlation that improved T cell frequency is associated with HBs antigen clearance. So that's what so those are some details about the HBV-specific T cell response, but now I want to talk about the mechanisms that are holding it back, that have allowed us to characterize it as weak or exhausted. And so there's a couple of different general concepts that are related to T cell exhaustion in hepatitis B. One is that it's a hepatotropic virus. So the liver is a tolerizing organ. It's a bad place to prime T cells. It has poor antigen-presenting cells in the hepatocytes, the L-sex, suppressive dendritic cells, macrophages, a lot of regulatory cytokines like IL-10, TGF-beta, PD-L1 expression, enzymes degrading essential amino acids, and it's also hypoxic. So it's really not a good environment for T cell priming. And there's also the high antigen burden that we see in a lot of the hepatitis B patients. They have high viral load, many of them have high antigen load, and in some cases the HBs antigen can be up to 1 milligram per mil. And we're just starting to get a better handle on the correlated antigen that's in the serum of these patients. And this could lead to constant presentation, either within the liver by hepatocytes or potentially in the periphery, leading to repeated activation of T cells, leading to their exhaustion. So what impact does this actually have on the T cell response? And these are the specific mechanisms really suppressing HBV-specific T cells. And probably the best described thus far is the inhibitory receptor expression. So in particular, PD-1 is the dominant inhibitory receptor expressed on HBV-specific T cells, but they're often co-expressed with one or two or more other co-inhibitory receptors. And just like the T cell phenotype that I showed a few slides ago, what we see is that these inhibitory receptors are upregulated to a greater extent in the liver compared to the blood. So again, compartmentalization of function. There's also a nice paper this year showing that there are significant metabolic defects in the HBV-specific T cell response, in particular related to mitochondrial dysfunction. And this would impair T cell functionality, and adding antioxidants into these co-cultures could restore T cell functionality in chronic HBV patients. In addition to these two mechanisms, there's also active elimination of T cells. So they're very hard to detect in general, and one of the reasons why is that many of them can be deleted, particularly in high viral load patients. And so it's been described that T cells can undergo activation-induced apoptosis. And if you block this pathway seen in the black bars here, you can increase T cell frequency. There's also evidence that HBV-specific T cells upregulate death receptors that are the ligand trail is produced by NK cells, so you can have NK cell-mediated depletion of HBV-specific T cells. So these are at least 4 different mechanisms specifically inhibiting the HBV-specific T cell response. But on top of this, we also have general mechanisms of immunosuppression operating in chronic hepatitis B patients. And one of these that was recently described is the increased frequency of myeloid-derived suppressor cells in chronic hepatitis B. These cells produce arginase and IL-10 that both in combination can suppress HBV-specific T cell function and expansion. There's also free arginase present in the serum of chronic HBV patients. And this often correlates with ALT because arginase is an enzyme within hepatocytes. It's released from hepatocytes. It depletes the essential amino acid arginine, which T cells need. So when you replace arginine in the culture, just like all the other experiments, you can enhance T-cell proliferation and expansion. And then lastly, out of these nonspecific mechanisms, both regulatory T cells and regulatory B cells have been described in hepatitis B. And these correlate generally with ALT and produce IL-10, which leads to a suppression of the HBV-specific T cell response. So we've come a long way in understanding the frequency, the function, what's holding these T cells back. And so we have a very good understanding of the T cell response now, and it's leading to development of new therapies. And every time I give this talk, one of the questions at the end comes to, what about the B cell response? And Peter Revel was up here yesterday talking about the ICE HBV initiative, and so I'm the co-chair for the Immunology Working Group. And so one of our main objectives was, or unmet needs, is we need more information about B cells. I mean, the point is that vague because we have ultimately that little bit of information about them. And I found that you could do pretty much all the reading for the B cells in hepatitis B in a single day. So I put them all up here. You're welcome to read them. But the way that we've historically measured the B cell response in hepatitis B is by looking at antibodies in the serum. And so some cutting edge data from 1979 shows us this kind of classic response to, I would say, probably most acute viral infections, where you have this situation where you have high antigen load, so you have more antigen than you do antibodies. You start to get clearance of the virus and the antigen. You generate immune complexes, and then ultimately you get clearance of the virus with free anti-S that's detectable in the serum as an indicator of resolution of disease. And these immunocomplexes are found in greater than 90% of acute patients, and you can find memory anti-S specific B cells by ELISpot in both resolved patients and vaccinated patients. The data from chronic HBV patients is a little more complicated. Most of the chronic patients are anti-core positive. Some become anti-E positive. And memory B cells thus far have been pretty difficult to detect in chronic HBV patients. And ultimately, there's also fewer immune complexes in chronic patients. Although when they are described, they're generally described in this immune active phase of disease, which interestingly correlates with data I think that was actually published after 2000, so cutting edge stuff for hepatitis B that shows that the immune active stage of hepatitis B is also correlated with an activated B cell profile. There's also evidence that some of the subclasses of IgG are better associated with control. But I would say that this is still quite top level, and it's recognized by many people working on immunology of hepatitis B that we need a lot more information on these B cells and what they're actually doing during disease. So with the next few slides, I want to focus on the innate immune response, and we know more about innate immunity in hepatitis B than we do about the B cell response, but still it's relatively less than what we know about the T cells. And one of the reasons why I would say that it's arguably more complex than the T cell response. We know in the T cell response we have the CD4, the CD8 T cells, different lineages, differentiation states of these, but ultimately in the innate arm of immunity we have multiple types of effector cells. So it's the NK cells, the MATE cells, the gamma delta T cells. And MATE cells have incredible diversity based on their KIR receptor expression profiles. Gamma delta T cells have multiple subpopulations based on their TCR profiles. MATE cells have many different specificities and diversity, but then also have myeloid cells on top of the effectors. So you have monocytes and macrophages, which each have their own subpopulations. You have dendritic cells with multiple subpopulations. And so these are just the hematopoietic cells, and this is not talking about the non-hematopoietic cells like the parenchymal cells, like hepatocytes or endothelial cells or stellate cells that all contribute to some level of innate immunity. And it's also important to remember that the innate immune response in chronic has mainly been studied in the context of the pathogenesis of chronic hepatitis B. And we have very limited data in studying or comparing the innate immune response from acute to resolved to chronic, like has nicely been done for the T cell response. It's also important to realize when we study the innate immune response that vertical transmission accounts for a majority of the infection, majority of the chronically infected patients. And so essentially there is no acute response to HBV because the virus is always present. and the innate immune system is actually exposed to HBV before birth. So I was part of a study that was published a couple of years ago where we looked at the monocytes in the cord blood of babies born to HBV-positive mothers or comparing them with uninfected mothers. And even in the cord blood, we could detect monocytes that had already internalized HBV surface antigen. And when we compared the transcriptomic profile of these monocytes to either the monocytes of healthy monocytes from uninfected cord blood or to adult monocytes, you can clearly see that the monocytes coming from HBV-infected cord blood clustered more closely with adults, suggesting that there is already maturation of the innate immune response, at least before birth. And so how does this alter innate immunity? Well, looking at the myeloid cell function in chronic hepatitis B, dendritic cells have been studied the most. Dendritic cell function has been shown to be highly variable, either inhibited, activated, restored with nucleoside analogs, and it's made the data difficult to interpret. But monocytes have been studied to a lesser extent. We've shown that monocytes can actually internalize and retain depots of HBV surface antigen in vivo. And despite being loaded with the HBV surface antigen, they didn't have any defect in their ability to stimulate T-cell proliferation. And other groups have shown that their ability to stimulate cytokines is still intact as well. And one of the caveats of studying the myeloid cells in chronic hepatitis B is that these cells are short-lived. They spend 1 to 3 days in the circulation compared to a T or B cell, which could be around for years or potentially decades. So these myeloid cells are going to be highly susceptible to environmental changes, probably have very dynamic responses, and ultimately it could be also impacted by bone marrow alterations. In terms of the effectors, the NK cell response has been studied the most in chronic hepatitis B, and it's been found that the NK cells, their ability to produce interferon gamma is suppressed, largely related to an IL-10 production, particularly in patients with hepatitis. Also, coming back again to this idea of compartmentalization, location matters. And so in terms of the innate immune response, innate cells dominate the immune composition of the liver. And this is something that's important to keep in mind because when we look at the blood and you look at the T and B cells, they comprise about 80% of the peripheral blood lymphocytes, whereas the innate effectors like NK and gamma delta and mate cells make up only about 20%. Whereas if you look in the liver, this ratio is almost completely inverted, and innate effector cells dominate the intrahepatic compartment. Myeloid cells are also altered to some extent. They have an expansion of this double positive CD14-16 monocyte population, maybe not as dramatic as the lymphocytes, but all this has pretty significant implications because, as you're probably all aware, a lot of the new immunomodulatory drugs for hepatitis B that are coming down the pipeline are orally delivered pattern recognition receptor agonists that are absorbed through the gut and go to the liver to stimulate innate and antiviral cytokine production. So it's imperative that when we're studying the innate immune response, we have to understand how these cells in the liver are responding to these drugs so that we can maximize their antiviral efficacy and minimize the toxicity associated with inflammatory cytokine production. So with that, I'll come to kind of a brief summary of these different topics. So the HBV-specific T cells, I think now we have pretty good data that they're not significantly different across the different phases of disease. The frequency is similar and the functionality is similar. The repertoire, it does vary by genotype, by the HLA profile of the population, as well as compartmentalization within the patients. And there are multiple specific and non-specific mechanisms that are maintaining the exhausted state of these T cells that are currently trying to be targeted for immunotherapy. The B cell response, the one slide I did present on it, this is pretty top level data. There's a lot more work that needs to be done. There's evidence that memory B cells are detectable and resolved in vaccinated patients, but so far it's been pretty difficult to detect them in chronic patients, and not a lot of people have looked. And better tools, there is some data coming on the horizon using better tools, and I think we'll see more about this in the next 1 or 2 years. There is good evidence now that the B cells have an activated profile during immune active disease, but we don't really know what that means. So the story of innate immunity is really key. So one is that we needed to enhance T cell immunity, especially like therapeutic vaccination, antigen presentation is key. They have significant antiviral potential if properly harnessed with these pattern recognition receptors. But effector cell function is reduced in chronic HBV, so the NK cells produce less interferon gamma and can actually negative regulate T-cell immunity, such as the MDSC and the NK trail. And so there's kind of a mixed bag of responses here with the innate immune response, and I think that's kind of indicative of its dynamic state, the fact that it's not long-lived. So with that, I will end, and before I do end, I want to make a couple of big, small advertisements, I would say, because we have a big year for us in Toronto, and I'm going to stand up here and I'm going to try and invite you to Toronto while we're sitting on the beach in Hawaii. I know it's not an easy task to do, but it is June, so it's a beautiful time in Toronto. But we're organizing the Global Hepatitis Summit. It's the 16th International Symposium on Viral Hepatitis and Liver Disease. We have outstanding faculty confirmed for clinical science, basic science, public health. We have an EASL and ASLD co-sponsored symposium on viral hepatitis and HCC. An ASLD, ALE, and CASL sponsored symposium, a Pan-American symposium on viral hepatitis and indigenous populations. We've partnered with the World Hepatitis Alliance to really get public health out in the exhibition space, a lot of programming in No Hep Village. Registration is open. I urge everybody to check out the website. It's going to be a fantastic meeting in June. I hope we all see you there. And then on top of that, I'd also like to invite everybody to come to our 5th International HBV Cure Workshop. So this will be the 5th year we've had this. It's always right before the ASLD meeting. It's a one-day symposium where it's a really focused meeting to update on the preclinical and early clinical stage drugs with huge chunks of time devoted to interaction and discussion. So with that, I'll thank everybody for their attention and happy to take any questions.